Nature is full of secrets, some beautiful, some dangerous, and some surprisingly healing. Bee venom, the same substance that causes a painful sting, may also hold the key to fighting one of humanity's most aggressive diseases, breast cancer. Bee venom has been feared for centuries, but new research shows it could one day help save lives. Bee venom, also called apitoxin, is a clear, bitter fluid produced by honeybees. For centuries, traditional medicine has used it for arthritis, inflammation, and even nerve pain. What makes bee venom truly remarkable are its components. Scientists have identified more than 40 active substances inside it, enzymes, peptides, and proteins. Among these, one compound stands out, melittin. Within the complex cocktail that is bee venom, one component stands out for its sheer abundance and potent activity, melittin. This powerful molecule makes up about half the dry weight of the venom, but what is it? Melittin is a peptide, a short chain of just 26 amino acids. Its structure is deceptively simple, yet it holds the key to its profound biological effects. It's an amphipathic peptide, meaning one end of the molecule is attracted to water while the other is drawn to fats. This dual personality is precisely what makes it such a formidable weapon at the cellular level. This extraordinary power lies in its ability to wage war on the very boundary of a cell, its membrane. Melittin acts as a cytolytic agent, meaning it can break cells apart. When it encounters a cell, its fat-loving end irresistibly inserts itself into the fatty lipid bilayer. Once anchored, multiple Melittin molecules cluster together, collaborating to create a catastrophic breach. They assemble into a structure known as a toroidal pore, effectively punching a stable water-filled tunnel straight through the cell's protective wall. This breach causes a complete structural failure, disrupting the cell's delicate internal balance. For a healthy, normal cell, such a violent intrusion is incredibly harmful, leading to cell death. So how could this destructive force possibly be harnessed for good? The secret lies in the subtle yet critical differences between healthy cells and their malignant counterparts. Researchers have discovered that cancer cell membranes are often fundamentally different. They tend to have a higher negative electrical charge on their outer surface compared to healthy cells. The positively charged melittin molecule is therefore naturally more attracted to cancer cells, like a magnet finding its opposite pole. Furthermore, the membranes of many cancer cells are more fluid and less organized, making them an easier target for melittin to penetrate. This selective attraction turns melittin into a microscopic guided missile. Once the pores are formed, the cell's defenses are overwhelmed. The carefully maintained balance of ions is lost in an instant as calcium floods the cell. This sudden calcium shock acts as a critical internal signal, triggering apoptosis, a process of programmed cell death. It's the cell's own self-destruct sequence, a clean and contained demolition that prevents the release of harmful substances into the surrounding tissue. For cancer cells, which already grow chaotically, this induced self-destruction is a fatal blow. This inherent selectivity is the holy grail for cancer researchers, but it is highly dependent on concentration. Finding the precise therapeutic window, a dose high enough to kill cancer cells but low enough to spare healthy ones, is a key challenge. But perhaps melittin's most exciting potential lies not in acting alone, but as a powerful ally to existing treatments. Many cancers develop resistance to chemotherapy by evolving molecular pumps that eject the drugs. Melittin offers a brilliant workaround. By punching holes in the membrane, it creates a new entryway, allowing chemotherapy drugs to flood the cell and bypass these defenses. This synergistic effect means that lower, less toxic doses of chemotherapy could be used, drastically reducing side effects and making treatments more effective against even the most stubborn drug-resistant tumors. In the global fight against cancer, scientists are searching for novel weapons in unexpected places. In 2020, a team at the prestigious Harry Perkins Institute of Medical Research in Australia turned their attention to a remedy with roots in ancient medicine, bee venom. Led by Dr. Chiara Duffy, the research sought to apply modern molecular science to an age-old question. Could this potent natural substance truly combat one of our most formidable diseases? Their investigation would provide rigorous evidence for what folk medicine has claimed for centuries. The team's focus was laser-sharp, targeting two of the most aggressive forms, triple-negative breast cancer and HER2-enriched breast cancer. Triple negative is particularly insidious because its cells lack the three common receptors that many current therapies use as a target. It's a cellular fortress with no doors for conventional drugs to enter, making it notoriously hard to treat. HER2-enriched cancer, meanwhile, has an overabundance of the HER2 protein which fuels its rapid, uncontrolled growth. While targeted treatments exist, the cancer can often develop resistance. To test their hypothesis, scientists sourced venom from honeybees in Perth, Western Australia. In the lab, they applied controlled doses of this whole venom directly to cancer cell cultures. But they went a step further, isolating Melittin, the venom's primary active component, and even created a synthetic version. This was crucial to determine if Melittin was the true cancer-killing agent and if it could be manufactured at scale. They applied these compounds directly to the cancer cells and a control group of healthy cells, measuring how long it took to act, how many cells survived, and how healthy cells responded in comparison. The results were astonishing. The researchers observed that Melittin was literally punching holes in the cancer cell membranes, causing them to rupture and die. This destructive process was incredibly rapid. Within just 60 minutes, Melittin significantly reduced the survival of aggressive breast cancer cells, effectively destroying them with remarkable efficiency. Even more promising, the same concentration had minimal effect on the healthy cells, suggesting a highly targeted toxicity. But that wasn't all. The researchers discovered a second, equally vital mechanism. Melittin also disrupted the chemical messaging pathways that cancer cells use to grow and divide, shutting down the command center for cancer proliferation. In some cases, the venom completely destroyed the cancer cells while leaving nearby healthy breast cells largely unharmed. Even more exciting, Melittin didn't just poke holes in membranes. It also interfered with critical cancer cell signaling pathways like EGFR and HER2, proteins that drive tumor growth. By blocking these signals, Melittin attacked cancer cells from two angles, physically breaking them down and chemically shutting off their growth instructions. This dual action makes Melittin an incredibly promising candidate for future cancer therapies. Scientists also tested Melittin combined with chemotherapy drugs and found that the venom helped deliver the drugs faster and more effectively into cancer cells. So what does this mean for cancer treatment? The true potential of Melittin may not be as a standalone cure, but as a powerful ally to our current cancer-fighting arsenal. Experts envision a future where it's used in combination with established treatments. The idea is one of synergy. Melittin could act as a sensitizer, effectively priming cancer cells for destruction. By disrupting their membranes, it could make them far more vulnerable to chemotherapy drugs, or prevent them from repairing the DNA damage caused by radiation. It's not just adding another weapon, it's about making every other weapon in the oncologist's toolkit significantly more potent. This synergistic effect has profound implications for patients. Conventional chemotherapy is a brutal systemic treatment that often brings debilitating side effects, chronic nausea, hair loss, and a compromised immune system. By enhancing the efficacy of these drugs, Melittin could allow doctors to administer them in smaller doses. This isn't just a minor adjustment, it could fundamentally change the patient experience, transforming a grueling ordeal into a more manageable journey, improving quality of life and allowing the body's own defenses to remain stronger during the fight. Nowhere is this hope more palpable than in the fight against triple-negative breast cancer. This aggressive disease gets its name because it tests negative for the three most common receptors that fuel breast cancer growth: estrogen, progesterone, and the HER2 protein. Without these targets, many of our most effective therapies are rendered useless, leaving patients with fewer options and a poorer prognosis. A new agent that can specifically target and kill these elusive cells would be a monumental breakthrough, offering a lifeline where one is desperately needed. If Melittin can make a real difference here, it would change countless lives. Of course, the path from a promising compound in a lab to a medicine in a clinic is fraught with challenges. What works in a petri dish doesn't always translate to the complex human body. And with a potent compound like Melittin, the primary challenge is precision. Unleashed in the bloodstream, it's an indiscriminate destroyer. The key is getting it to the tumor, and only the tumor. To solve this, scientists are turning to nanotechnology. They are engineering microscopic carriers to encapsulate the Melittin, shielding the body from its effects. Like a Trojan horse, These nanoparticles circulate harmlessly until they reach their target. They can be designed to accumulate in the unique environment of a tumor, where they break down and release their deadly payload exactly where it's needed, minimizing collateral damage and maximizing its power. There are also ethical questions. Large-scale use of bee venom raises concerns about sustainability and animal welfare. That's why most current research uses synthetic Melittin, created in labs without harming bees. To recap, bee venom contains Melittin, a small but mighty compound that can rapidly destroy aggressive breast cancer cells in lab tests. In less than an hour, it's been shown to kill cancer cells while sparing healthy ones. The promise is huge, but it's still early days. Human trials, safe delivery methods, and long-term studies are all needed before Melittin can become a real treatment option. The discovery of bee venom's potent effects on cancer cells is promising, yet it's important to approach this new frontier with careful optimism. Bee venom is not yet a cure, but it is a powerful clue. As research continues, Melittin may become part of a new generation of therapies that combine natural compounds with advanced medicine. What do you think about bee venom as a potential cancer treatment? Is it the miracle cure we've been waiting for, or just the first step on a long scientific journey? Share your thoughts and keep the conversation going. Thank you for exploring this fascinating discovery. As we continue to unlock nature's hidden cures, who knows what other secrets are waiting to be discovered? Until next time, stay curious and stay healthy.